Well, so yes, as you said, I had this title, but after seeing John's lecture yesterday, I think I should rephrase it to the simulating the pathway of compaction of centromeric units in holocentric species. And so I'm going to talk about molecular dynamic simulations, polymer simulations, trying to analyze how centromeric units are arranged in specifically holocentric species. And holocentric species actually was already introduced yesterday as well by Peter. So the main feature is that instead of having a monocentric, so a single constriction of the chromosome with microtubules binding to it, we have microtubules binding all along the side of the chromosomes. And then during segregation of the sister chromatids, they're separated parallel. So these are holocentric chromosomes. And we wonder how the centromere can arrange itself in so many different forms. So the first thing we're wondering, so actually in some time ago, the centromeres were already analyzed and described as being formed by centromeric units. So it's not a single structure, but it's formed by many smaller structures that are called centromeric units that would bind after to kinetochores and then to the microtubules. So this is valid even for monocentric chromosomes. And so the centromeric units, they could be called by being composed of kinetochore proteins, but the entire kinetochore complexes is also containing these centromere specific histone 3, CNA3. They are part of nucleosomes. So some of the nucleosomes in chromatin contain these special CNA3 histone, and some are not. So some of the nucleosomes will be called centromeric units and to differentiate from the others. And there's not only one in the chromosome, but several, and they can be distributed all along the chromosome, which is what for the holocentric chromosomes, or in a small region as for the monocentric chromosomes. And there can be also other nucleosomes in between them. So if we can have normal nucleosomes in between a few centromeric units. And just to differentiate them, so from monocentric and holocentric, where are these centromeric units found? So it's different in interphase and in metaphase. So for monocentric chromosomes in interphase, there is a broad signal for this CNA3, but they are single signals for each chromosome, and then they are observed very sharply in the metaphase. And for holocentric, there are actually many centromeric units distributed all along the nucleus, and there are many more than just the number of chromosomes. But then to metaphase, they come to form this line. So what is very intriguing is how these many centromeric units come to arrange into this line. So this is the problem that we addressed. So during the compaction, so during the cell cycle, the centromeric units, so intercalating between normal nucleosomes, would come to form a line. They would arrange and come together, forming this long line in prophase, and then in metaphase, it would be even more compacted into a shorter line together with the kinetochores of the other proteins of the kinetochore complex and the microtubules. So what we are addressing in this presentation only is just this part of the cell cycle. So going from interphase to prophase, how do they come to this line? And the method that we use to approach this issue, this problem, is molecular dynamics or polymer dynamics. And the model that we are using is saying that the chromatin is actually a polymer formed by nucleosomes, and each nucleosome will be one bead. So it's a beads on a string model, and each bead is connected to the other by harmonic springs. So there are forces in this chromatin fiber to simulate its motion inside the nucleus. One of them is the harmonic force between the beads. The second of them is that they are allowed to bend. And the third that there is a repulsion force between them, but we also consider that there is topoisomerases, and sometimes the chromatin fiber is allowed to cross itself. And the third force is a damping force mimicking the solvent surrounding it. So it's not a polymer in space, but it implicitly considers water and other solvent molecules. And to go to study the compaction, we consider it's being done by loop extrusion. So I'm not going into many details here because it's already been touched in many ways by the previous lectures. I just want to say how to, so in our simulations, I simply consider there are pairs of nucleosomes that are differently bound during the simulation. So in the first step, these two are bound together, and then the second step, these two are bound together by a harmonic spring. And then the third step, different nucleosomes are bound together, and then increasing the distance, and for some time, then the SMC complex will be allowed to be released. So it has a lifetime that we consider. And this compaction by loop extrusion, we know that it's able to lead from a sparse state to a compacted state. So if the more SMC complexes we consider, or the more time that these are bound to the chromatin, they can form this compacted state, which is formed by loops side by side. And they're side by side because most often the SMC complexes cannot traverse each other. I know that there are some studies saying that it's possible, but in these simulations, I consider that they are always blocking each other's way. So they are always compacting to this kind of loops that are side by side or nested. And at least what I consider for the centromeric units is that they act as barriers or more as anchors to the SMC complexes. So when they are extruding the loops, whenever they find a centromeric unit, it stops there. And this simply concept would bring the centromeric units to form a line and being the center of this chromosome and loops emerging type from it. So it's just an idea that we wanted to check, and I performed this molecular dynamics to see if it's true. So I have a very simple example here of 1 megabase chromosome with 25 centromeric units distributed randomly. And applying the loop extrusion motion, then we can see that the centromeric units indeed brought together forming this line. So it's just a simple concept that we think may be the mechanism behind the motion, so the arrangement of the centromeric units from interface to prophase. So this is a very small chromosome, and just to give you an example of a higher or more realistic chromosome, this is a 20 megabase example. They are found in C. elegans, so holocentric species, and they have, with chip-seq experiments, they have seen the distribution of CNH3 in chromosomes, and there are probably like 100 centromeric units. So this is what I put here in this non-compacted state, randomly distributed. And then after compaction, we come to models like this. So we see still the line of the centromeric units and loops coming out of it, and the centromeric and the SMC complexes binding along this line as well. And one of the characteristics of this compacted model is that these centromeric units are brought together side by side in a linearly way. I mean that they follow the genomic position. So the centromeric units that they are close in the genomic sequence will be also close in the structure. And so one further experiment that supports this mechanism is that cleansing, so subunits of the SMC complexes are indeed found to the side of the CNH3 So these are centromeric units in forming a line, and indeed close to it, there are the SMC complexes. And so we propose that other experiments could be done. So using the model that we have, we could predict how would be a high C matrix, and we see that besides the main diagonal, it also forms in this compacted state. It would also appear as small squares resembling dots formation with these highlighted dots here, signaling that we have these regions coming together very often, which would be the regions where we find the centromeric units. And also possibly we could, with oligo probes and fish experiments, so color different regions and analyze and see that it's really linearly arranged in this compacted structure. Well, and I guess I was very brief or not, but this was everything that I had to show today. So I'd like to thank again the possibility to present here. And I need to thank Andreas Ruben and Kai Schobel for bringing up this subject and with the experiments of microscopy, and Martin Marshall, who is my host in IPK, for the possibility and support with the simulations. Well, and thank you very much for your attention, and thanks.
Thank you, Amanda, and happy that we could hear you and listen to you. So Daniel has a question.
Yes, hello Amanda. Thanks for the talk. So in your simulation, and you can see that your boundaries, okay, are found in the middle of the big chromosome somehow in the center, and it seems that actually the holocentric modules are more at the border of the surface of the chromosome. So do you have any explanation for that?
Yeah, actually, if I can share this slide, actually from another presentation, if you don't mind. So these are again holocentric chromosomes, and besides being found in the side of the chromosome, there is also forming this groove. And what we think this is coming from is that with after forming this centromeric line in metaphase, we consider the existence of the kinetochore complexes bound to it. So if they are bound to one side, the same SMC complexes can only be to the other side. And if they are to the other side, the emerging loops can also be to the other side. So we have loops coming out in this direction. But the loops are quite flexible, so they can occupy the space surrounding it. And if we fill it with loops, we can then see that they form this kind of roof around it. But they are mostly to the side of the chromosomes. Okay. Would that answer your question?
Yeah, it's perfect. Yeah. And I have another question regarding these regions.
I'm sorry.
Are they covered by HP1 also, these regions? And is HP1 still there during this prophase formation?
I couldn't say. I'm sorry. I didn't consider this in my simulations, but I would be happy to consider them in the future.
Okay, thank you.
We have a question by Eloise Muller. So what would you think is making the difference between interface and prophase when compaction started in your centromeric units to trigger this block of SMC, which does not seem to exist in interface?
So in interface, I'm sorry, I was left with the other presentation. So in interface, they are spread off the nucleus, and so we know from the other organisms that SMC complexes are there and acting and making loops, but there are different types of condensins, and they come in different phases of the cell cycle. So what I would expect and what I've seen in previous studies is that this compaction comes from condensin too. So perhaps it is acting at different stages of the cell cycle that wasn't present in interface. So I guess what would trigger it would be SMC complex more active than before. Does that make sense?
Yeah, but in your simulation, so where do you?
In my simulation, it's just the beginning of the loop extrusion. So in interface, I wasn't considering there was any loop extrusion. And then when the loop extrusion starts, they began to compact and they come to a line.
Okay. But I mean, am I wrong if I say that there is evidence there is loop extrusion in interface?
No, yeah, there should be. I was a very simplified model, so I was considering any, but perhaps I could consider perhaps less condensings or other type of condensing.
So concentration of condensing. Okay.
Yeah, could
so
be,
actually, yeah. I had a related question. So here it looks like you form a flexible fiber which has a persistent length. So do you know what defines actually the persistent length of this, this prophase structure?
And the one
in your simulation, obviously. Yeah.
Yeah. Well, so this more complex. So this fiber. So you mean this prophase fiber with the loop?
So typically here there is a curve, etc. So so here, I mean, some of the parameters of your simulation will give a shape more rigid or more flexible to these things. So do you have any hint or any idea of what could it be? It could be the concentration again.
No, I didn't consider this, but it's a very interesting question. I think that we should consider also the presence of other chromosomes if you want to study that.
Yeah, sure. Yeah.
And in this case, was just one. So, but it's interesting. I will consider it.
Okay, very last question. We are very late, so we can take advantage of that. So is there any evidence by Jonas Poulsen, is there any evidence that beside your simulations, obviously, that the centromeric units act as boundaries?
First, hi, Jonas. And well, it's been suggested that other proteins should act as boundaries. And there are other papers suggesting that centromeric units could be acting as boundaries. And I think mostly just because perhaps of the size, perhaps can be a stereo constriction. Because if you imagine the entire kinetochore complex or other proteins binding to it, and that the chromatin fiber should go through the SMC complex, we can only imagine that proteins that bind to the chromatin fiber could affect the motion of the SMC complexes. But I will start from the fact that other proteins are already found to be barriers.